So good morning, everybody. Today, what I'd like to talk to you about is our efforts on cracking the morphogenetic code. And to think about why this morphogenetic code is important, we need to think about how remarkable bodies are in terms of editing their own shape. And all of these problems, birth defects, which are errors of initial patterning, traumatic injury, cancer, or the defection of cells from the anatomical plan, and even aging, have one fundamental thing in common. What they have in common is a failure of the ongoing control of complex anatomical structure. And so in embryonic development, all of us start life as a collection of early blastomeres, and then through embryonic development, we acquire this tremendous shape. So here you see this cross-section through a normal human torso. And what you notice is an amazing increase in the amount of anatomical complexity to go from here to here. Now, these processes can sometimes go awry, in which case you may have a tumor that still contains individual tissues, hair, teeth, skin, and so on. So cell differentiation is crucial, but not sufficient for obtaining this kind of pattern. And so we really need to understand where the information is that drives complex patterning. Past embryonic development, we have a number of organisms that are telling us a proof of principle that full regeneration as adults is possible. Here are planaria, which are flatworms that have this amazing property. You can cut these guys actually in any direction that you want. They will regenerate. Every piece of these worms will regenerate exactly what's missing, no more, no less, and they stop when they're done. Not only that, we showed recently some work that these planaria actually keep their memories through complete brain regeneration. They are able to resorb their tumors when regeneration is activated, and there's actually no sign of aging at the level of the organism. So individual cells senesce and die, but the animal continues to regenerate itself, and so is practically immortal. So this is the result. All of these amazing features are the result of this animal having incredible pattern control over every piece of the organism. Now, regeneration is not only for so-called lower organisms. We have vertebrates, such as these salamanders, that can regenerate their limbs, their eyes, their ovaries, their jaws, their hearts, and so on. And of course, even deer, which are adult large mammals, can regenerate a tremendous amount of bone, up to 1.5 centimeters of new bone growth per day when the antlers are being regenerated. And of course, the human liver is known to be regenerative. So fundamentally, this kind of process where a complex structure is amputated and then regenerates is asking us to solve several problems. First, the relationship of pattern to the genome, that is, where is the information stored and processed such that we are able to restore from damage. And then as engineers, we ask the question, how far can we push shape change? That is, what patterns are possible to obtain given a particular genome? So these kinds of processes, both standard embryonic development, where everybody starts at the same starting state and then reliably reaches one of these complex morphologies, or in fact, very plastic events such as this, where, for example, you can take a tail of a salamander, attach it surgically to where the limb goes, and over time, this structure will remodel itself into a completely new structure that's more appropriate to the large-scale anatomy. So this, of course, is a process that starts in a very aberrant state and yet manages to reach the correct final goal. So how much of this is locked in or fully determined by the DNA, and what are the evolutionary implications of this kind of plasticity? So we think of this as a closed-loop pattern homeostatic process whereby a set of gene regulatory networks create proteins, these interact, then through emergent processes you have complex shapes, but there's a feedback loop where both at the level of gene expression and at the level of physics there is correction going on that continuously tries to get back to this anatomical state despite unpredictable perturbations. And there's a fundamental theorem in cybernetics and control theory which says that every good controller of a system, and this is an extremely good controller of this system because, for example, every regenerating limb is exactly the same as the original limb. Every good controller of a system must be a model of that system. And so if that's true, then there's a counter-paradigm idea that we could follow, which is that bodies might be able to store this model of the pattern and thus be able to represent an anatomical set point. That is, every kind of homeostatic process has to fundamentally store a set point towards which it fixes. And we call this set point the target morphology. And if that's true, then we could do several things. We could target this control system, we could rewrite the set point, and then we could let the cells build towards that plan. And in our Allen Discovery Center at Tufts, this is what we've been working on. I want to show you why this is important. This is just to briefly mention, two very fundamental problems that are still with us even in the age of genomics. So here are planaria, and the thing about planaria are that they largely reproduce by fission and regeneration. So each worm tears itself in half, the head end regrows a tail, tail regrows a head, and now you've got two planaria. What this means is that in this process, they escape Weissman's barrier. That is, if they acquire somatic mutations somewhere in the body of the organism that doesn't kill the organism and happens in one of these neoblasts or stem cells, that mutation is propagated to the next generation. Now, this is not true for us. If you have a mutation in your arm, the offspring don't automatically get it. But with planaria, they accumulate mutations over time. And so despite hundreds of millions of years of this somatic inheritance where they accumulate mutations. In fact, the genomes are very messy. These worms are mixoploid. The cells don't even have the same number of chromosomes. So they accumulate all of this stuff. And despite that, after many millions of years, they still regenerate with 100% anatomical fidelity. So here we have an interesting distinction between what's going on with the genome versus what's going on with the anatomy. And in fact, unlike mutant lines, which you can find in other species, Drosophila, C. elegans mouse, you can get lines of species with various patterning problems. There's no such thing except our two-headed worms, which I'll show you in a minute. There are no patterning mutants in planaria. These guys are incredible at keeping their shape despite this destabilizing process. It is not well understood at all. The other issue that isn't understood is if we try to make a prediction of what happens when we take stem cells from a flat-headed worm and mix them in the same body with stem cells from a round-headed worm, we let them sit there, then we amputate the head. What kind of head shape will result? The interesting thing about this thought experiment is that despite all of the work on molecular genetics of stem cell differentiation, we don't have even the beginnings of a guess of what to predict here, because none of the existing models address a fundamental question. How do the cells know when to stop, and what pattern causes regeneration to cease? Okay, and this is simply unknown. And so these are the kind of issues that are required for us to understand. And so we really need to get at the algorithms. How do cells make these decisions? What are they measuring in understanding what shape to make and when to stop? So in the computer science way of looking at things, it's clear that there's a next revolution in biology coming. And so we thought about what key insights allowed computer science to drive a revolution. What was it about early computer science that really impacted every aspect of our lives? And I think two arguments can be made. That the first really important thing is the distinction between hardware and software, and a focus on the control of information, not just the materials. And so this is how people used to program computers. You really had to be down in the hardware. You were literally moving wires around. And then eventually through progress in the field, we got a machine code, which was still pretty close to the hardware, but certainly better than this. And so now we have these top-level object-oriented languages that allow you to think about the information flow and the computation of the problem and not really worry about the hardware as much. And I think that in biology, we are still very much at the machine code level. Everything we do is largely focused on understanding how to manipulate individual molecules, where I think what we really need to also understand is the algorithms and the decision-making among cells, and to gain control of the computation that living matter performs in making decisions such as when and how to regenerate or restore or repattern large anatomical regions. So if we really understood what was, how these things work, we could not only defeat these major biomedical problems, but we could also, of course, create new biological constructs and also make important additions to robust robot technology. So what I want to do now is give you two updates on our work at the center that address some of these issues. And these are the members of our new center. Two of my colleagues, Kelly McLaughlin and Dave Kaplan, are here with us. And the two stories I'd like to update you on first has to do with our efforts to identify novel inputs into patterning. That is, what are the sources of information for patterning? And this involves a story recently that came out of our center on the brain as the first functional organ in development. What we basically found is that, and here you're looking at a very early frog embryo. This is about two and a half days of development, so very, very early on. What we found is that the very early brain in this animal, long before there's any behavior or anything like that, it is absolutely required for patterning events throughout the animal. So for example, you can see what happens here. Basically what we did is amputate the early brain, and in the absence of the early brain, the animal continues to develop, but we see really characteristic defects in innervation, in muscle patterning, and several other systems. This brain is having an important patterning role at considerable distance, and then what we found was that we could actually largely restore, not entirely, but largely restore both the muscle and the peripheral innervation patterning by judicious misexpression of appropriate ion channels and certain neurotransmitters. So there's this influence of the brain that's required for patterning to proceed, and to some extent that can be compensated by interventions that we apply. And so this was really remarkable for a number of reasons. First of all, the brain appears to be the first functional organ. This occurs even before the heart starts beating. It instructs pattern at long distance, so every part of the embryo is actually receiving this influence. The brain does this while it itself is being constructed. We showed a couple of years ago that in determining how big the brain should be, it's receiving input from the rest of the animal. So the rest of the animal, its electrical properties are influencing growth and patterning of the brain. While this device is being put together, it is already functional to, in fact, in this sort of complex loop, to pattern the other organs. And the other interesting thing that we found is that the brain protects the body from teratogenic influences. And what I mean by that is that drugs that normally do not cause any sort of teratogenic problem, they don't affect development, in embryos without a brain, they cause terrible problems. So the presence of the brain is able to protect the rest of embryogenesis from specific teratogens. And so the next steps for this process is, of course, to try to use this assay in a gain-of-function way to see if brain implants can improve regeneration and repair in various contexts, to see if we can mimic brain activity with targeted interventions, and design better screens for understanding teratogenesis, because it appears to not be true that you can simply sort compounds into the classes of being a teratogen or not being a teratogen, because whether or not that occurs is really determined by what's going on physiologically in the embryo. The other thing I wanted to talk to you about is our efforts in non-neural bioelectricity. So the brain uses ion channels and electrical synapses known as gap junctions to drive the electrical activity that is the substrate of the software of the brain, the activity. And so here what you're seeing is an active zebrafish brain as the fish is going through its cognitive programs. And there are these efforts in neuroscience on neural decoding. So the idea that you should be able to read out these electrical patterns, and then from that, using some sort of computational pipeline, extract the information content. In this case, what image is this person thinking of? So it turns out that in the rest of the body, we have something very, very similar. That is, all cells, not just nerve and muscle, have ion channels. They communicate with each other through these electrical synapses. And what you're seeing here, this is an early frog embryo. This is a movie originally made by Danny Adams in our group. And what you're seeing here are all the electrical conversations that these cells are having with each other as they determine which ones are going to make which organs. And we think that the information encoded in these electrical states represent pattern organizational influences that are going to be required both to make the animal in the first place and then to repair it after damage. So it turns out that bioelectricity is not something that showed up when brains evolved. Lots of cells use electrical signaling. This is an ancient, well-conserved signal. Here you can see one single cell undergoing some very complex behaviors as it hunts for its food. And basically, it's the case that brains co-opted many of their tricks from far more ancient usage of electrical circuits throughout the body. So what did tissues think about before they became brains using this strategy? And what tissues thought about was their own shape. They thought about the shape of the host in which they located. They processed information, and there's a tremendous amount of parallelism between the way that electrical circuits are used to process information in the brain and in the rest of the body. Now, we developed molecular tools to be able to direct and characterize these states. And so here you see a normal pre-pattern in this early embryo, where each one of these little circles is one cell. That cell drives its voltage potential using channels in the membrane, and so you can see this complex pattern across the anatomical scale. Or you can also detect pathological states. This is some of our work detecting tumors as their aberrant signature of bioelectrical signaling before the tumors actually appear. So the way we functionally manipulate these networks, we do not apply electrical fields. What we do, and here's a typical cell network. This might be an epithelium or a set of mesenchymal cells, whatever. We have two basic options. We can control the electrical synapses, so we can either open or close the electrical communication among the cells using either genetic or pharmacological means. So we can control the topology of this network. So this is basically analogous to synaptic plasticity. Or we can go in and directly, using either optogenetics or other tools, directly control the voltage states of individual nodes in this network. And so our strategy to rewire these circuits is first to computationally model the whole circuit, and I'll show you how we do that, and then to try to decode their pattern semantics, and then to try to alter these patterns in vivo using either channel targeting drugs or light, and then to try to actually alter the outcomes of this patterning process that's driven by these electrical circuits. And in the past, we've had applications in birth defects, regeneration, and tumor reprogramming. I'll give you one simple example. This is attempting to induce regeneration in an animal that normally does not regenerate its limbs. So here's a frog. Normally, you cut off the leg. 45 days later, you either get a spike or nothing. And so we came up with a cocktail that basically resets the electrical properties of the stump to that of a developing early frog. And so what that does is it kickstarts the whole process. So you've got some toes, you've got a toenail. The leg actually ends up looking quite respectable here in terms of its patterning. And you can see here that it's both touch sensitive and motile. So a complex physiological state can kickstart all of the downstream patterning modules that it requires to build this leg. Note that we did not have to do a micromanagement of every piece of this. We didn't have to say, here's where the nerve goes, here's where the blood vessels go, here's where the bone goes, and so on. This is a very modular process. The same thing actually can be seen here. This is an early example of turning a portion of the gut into a complete eye. And this, again, is a very modular event. Once you induce an eye-specific state here, the system does the rest. The cells basically build this eye without us having to understand exactly how to microassemble a leg or an eye. And so one way that we're thinking about this is different from the sort of current assumption, which is that the DNA is often thought to be the software, and then the cell is kind of the hardware that reads it. I propose a different view, which is that what the DNA does is it specifies the hardware. The DNA tells you which ion channels each of your cells get to have. But once you have those ion channels, there are complex electrical dynamics that go on that are somewhat akin to information flow in things like flip-flops, where the physical structure is set, but the information is stored in the pattern of energy movement through the system, and that is how computer memory stores information. That these events here are up for grabs in an important sense, that we can go in and intervene at this level, alter the physiology, and reach outcomes that are not constrained by the underlying hardware. So one example of this from our work with planaria showed that we can cut off the head, we can alter the network topology of the resulting fragment, and as a result cause this thing to grow heads that are either flat, like this P. felina, or round, like this S. mediterranea, and literally get it to build heads that are appropriate to other species. Now, this is not only in terms of head shape. They also get the shape of the brain and the stem cell distribution appropriate to these other species. About 150 million years of difference between them, and this is despite the fact that the genomic sequence is untouched. There's nothing, there's no genomic reprogramming that goes on here. This is a purely physiological effect, and you can see that from the same genome, you can actually derive shapes of quite different species. And in fact, you can go further, and you can make flatworms don't even have to be flat. You can make flatworms that look like little spiky hedgehogs, or little cylinders, or in fact leave the plane of the sort of more or less two-dimensional body plane of this worm and produce these tube structures on top. So what's important from these examples is that the bioelectric network can redefine anatomy without having to alter the genomic sequence. And we then ask the question of, well, how about basic body plan? At least surely the number of heads an animal has is set by the genome. And so the mainstream way of thinking about this predicts that if you made a two-headed flatworm, which we can make by altering the bioelectric signaling between the two wounds, Then, if you leave it alone for a couple of weeks and you amputate this ectopic tissue and also the primary head, and you just take this normal gut and you culture that in plain old water, given the normal, the wild-type genomic sequence, what you're going to get surely is a single-headed worm. And if I'm here telling you about this because that's not at all what happens. What happens is that in perpetuity, if you keep doing this, once you've shifted the stable attractor of this electrical circuit to a two-headed state, this thing will continue to regenerate in water with no further manipulations, despite a normal genomic sequence. So we see here that a target morphology of this species, or the shape to which it repairs upon damage, can be permanently respecified by altering the information in this electrical pattern memory. Now, why do I call it a pattern memory? This is a very recent story that just came out from our group, which shows that what you can do is you can take worms that are perfectly normal in their anatomy, so head and tail. They're also normal in their molecular histology, so the head marker is here, the head marker is not in the tail, and so on. So there's nothing molecularly wrong that we can detect in this worm. And yet this worm is going to give rise to a double head, and this worm is going to give rise to a single head. Now, why is that? What could be different about it? And it turns out that what we can do is rewrite the electrical pattern in this mature adult animal, such that you can see here, this is a one-headed voltage pattern, this is a two-headed voltage pattern. We can change the electrical pre-pattern, but notice that this is not reflective of what's currently happening in the animal. This is a perfectly good one-headed, anatomically normal planarian. What this is a latent memory. This is an electrical pattern that will become active if the animal is cut later on. Until that happens, they stay one-headed and everything is fine. If they get cut, they consult this electric circuit and they end up making two heads. So the same physical body can store inside representations of distinct outcome future states that it's going to acquire once it is cut. So we think that there are significant parallels here between the way that electrical networks store memories and make decisions in neuroscience, and we're making some connectionist models towards that end. But the other very important thing is to make really clear computational models of how this works. And so we're in the process of doing that. This is a new paper that just came out with the next version of our modeling tool. Our modeling tool allows us to take realistic electric circuits from data that we know are from proteins that are operating in the worm and our physiological data. It makes fully specified computational models of regenerative process. And from those models, we're trying to derive state space models like this that explain why any given circuit has particular attractors that drive it to stable two-head, one-head, or in fact, no-head states. So this is an important effort for us to synthesize from molecular information to physiological tissue-wide information to full anatomy-wide understanding of where these large-scale features come from. And so the second story I want to talk to you about is how we drive these kinds of advances towards hopefully some sort of practical biomedical application. And the arc that we try to follow here is to identify the endogenous bioelectrical pattern that is involved with some sort of organ that we care about, to build a computational model of it, to test and refine the model, and then use that model to extract predictions. And when we can extract predictions, we can also identify candidate treatments or interventions that drive the system to a particular state. And then, of course, we want to demonstrate efficacy in some model system. So the application I want to show you has to do with the frog brain. And so here's the early frog brain being developed. And what was discovered is that the early frog brain has this very characteristic voltage pattern here. And in fact, and here it is. If you move across from left to right here, it's got these sort of depolarized edges and then this hyperpolarized bit in the middle. And when you expose this animal to various teratogens, what will happen is that you collapse this interesting gradient so that everybody is up here. When that happens, the brain is terribly malformed. You get these really bad malformations. But if you try to hyperpolarize this thing naively, let's say to here, every cell goes to here, this is just as bad. Okay, and it makes perfect sense because it's not simply that every cell is at the same voltage. You actually need this pattern for correct brain morphogenesis to occur. And so what we were able to do was to use this computational model to understand where the electrical differences between this middle portion and the edges are coming from, and then come up with a specific set of existing ion channel drugs, and these are drugs already approved for human use in various applications. But what the model does is it tells you exactly what you have to do to drive the voltage to a particular pattern that you want to see. And so when you do that, here's a normal frog brain. You see forebrain, midbrain, hindbrain. Here's, in the case of the teratogen, the forebrain is very shortened. The eyes are touching the brain. There's significant malformations here. And in fact, on top of this, you can apply a couple of the drugs, actually one at a time, that was identified by this model. And you basically rescue not only the anatomy of the brain, but also the gene expression and the IQ of these tadpoles. The tadpoles do have an IQ. You can measure their learning speed and so on. And they actually get their behavior back. The other really interesting thing is that this is not only for chemical teratogenesis. These are animals that are misexpressing a dominant mutation in Notch. So Notch is a really important neurogenesis gene, and when they have this mutation, you can see compared to a normal brain, you can see here that their brain is really quite screwed up. And so even on top of this really debilitating mutant phenotype, you can, by putting in appropriate ion channels, you can rescue, again, entire anatomy of the brain, molecular markers, and also their behavior. So this is an example of using and targeting these bioelectrical states to overcome either chemical teratogenesis or, in fact, a really dominant genetic mutation. So what we're doing now is trying to put together a predictive simulator that would allow us to take existing ion channel expression data from other workers, where we now pretty much know what ion channels every tissue in the body has. From our work on cracking the bioelectric code, we try to understand what VMEM pattern is desired. So let's say for brain patterning, for limb patterning, for all kinds of other anatomical features. And then what we would do is use the simulator as a way to predict and design cocktails of existing ion channel drugs that could be applied, leveraging the differential expression of ion channels throughout the body to try to rescue appropriate pre-patterns, such as this early face pattern where you get to see where the eye is going to go, the mouth, and so on. So the idea is that we're working on this kind of predictive platform for suggesting ion channel drug interventions for specific uses. And so one of the nice things about these models, and we also have some interesting machine learning approaches to make it easier to do that, once these kinds of networks are developed, they can be interrogated to find this kind of needle in the haystack. This is our work showing that in the transition from normal pigment cell to melanoma, there are very specific choice points that can be identified, that can be targeted to drive body-wide outcomes. And this is an example of using both machine learning and our bioelectrical modeling platform to identify specific interventions that will let us have really rare outcomes. And so to finish up, in summary, what I want to tell you is that endogenous bioelectric circuits control gene expression and anatomy, and that the activity of these circuits can be targeted. Their function can be rewritten to alter species default outcomes, either through patterning or through gene mutations and so on. That new computational and biophysical approaches are now coming online that will allow us to rationally target decision-making on the tissue and whole body level. And I think this opens many opportunities for addressing birth defects, degenerative disease, aging, injury, cancer, and so on. And I also think that the major transformative advances in this field require us not only to understand the mechanisms, the molecular mechanisms by which these things work, but also the computations that cells and more importantly tissues perform to make decisions about what they should be doing so that we can alter these decisions. So I would like to thank the people who contributed to this work, and of course the Allen Frontiers group for funding. And I leave you with a picture of our two-headed planaria, which have been called impossible objects because these guys arise from a genomically wild-type animal cut in plain spring water. So thank you. I'll take questions.
Michael, in your model of bioelectrical fields being the software on the nucleic acid encoded hardware, are you looking at what presumably is small gradient field effects on the actual cellular machinery to be asymmetric or polarized or make micro decisions to set up these larger morphogenic kinds of changes? How are you going about that, if at all?
Yes, yeah, absolutely. I don't have, just for reasons of time, I didn't bring all that stuff here, but we can talk about this afterwards. Basically, what happens is that in the model, small local changes percolate up. Okay, and so you do start with localized cell level changes that percolate up into large scale decisions where the cells coordinate with each other. One of the things the bioelectric layer is very good at is for coordinating cell activity across large areas. And so both in the case of brain development, cancer, some of these other examples, what happens electrically in one part of the body has a massive influence on another part. And so our goal is to understand in both directions. We need to know what's happening at the cell level, drill down, which we've done. We understand the transduction machinery to some extent, what genes are downstream. But also to go upwards and understand the large-scale decision-making that this enables. Because the things that we're talking about often cannot be decided on a single cell level. These networks are making decisions about things that are large, right? And so propagating that information up is crucial.
So, hey Mike, I love your work. It's so most of what you showed us could be explained by I mean, I couldn't agree more that the DNA is basically the set of tools and not a code for how to make an animal. And it's, so most of what you showed us could be explained by the fact that you could rewire the circuitry of a cell into a sort of a stem cell state, starting with the sort of the bioelectric aspects, which then feed to the nucleus, you change gene expression, and cells then reprogram themselves into stem cells, so in a regenerative capacity to recreate the pre-existing pattern, and then you basically have sort of tissue-wide morphogens and so on. It's classic developmental things. The only challenge to that idea would be the fact that you create this double-headed worm in a completely. So what do you think? I mean, this is related to Jeff's question. What do you think is happening there? Because the issue is that you have to create cells that are in a stem-like state in order to generate other cells and give rise to different types of these cells. But at the same time, they've got to sort of follow a different plan, so to speak. And it's not clear to me where that type of information, where that type of plan would be. And maybe you just didn't touch upon that.
Sure. Yeah. Right. I simply didn't go into detail, and we can talk about this offline. There's two important things in what you said. The first is the issue of stem cells, and I'll just talk briefly about that. It is true that, let's say, in culture with human stem cells, let's say mesenchymal stem cells, you can use bioelectrical properties to control specific stem cell differentiation. This is true. In our particular example, in the case of the frog, there really are no, that's not the case. The frog does, it's tissue renewal, so every cell makes more of itself. There's no reversion back to a stem-like state in the frog. In the planaria, it's sort of the opposite. Planaria have these adult neoblast stem cells that are like 30% of the animal. They're always there. And again, we have no evidence that we're making new stem cells. It's literally, the stem cells are doing their thing. It's, as you said, it's the plan that these stem cell progeny have to follow. And this is actually an issue for all of our processes, because simply having specified what cell types you have, right, is not sufficient. There's a next step, which is how do you organize all this stuff. So the worm is the model where we best understand it, and basically what we have now is a model in which the information, at least let's say a very simple example, head or tail polarity, is literally stored in the electrical circuit. I'll draw it for you afterwards, that you could literally draw the electric circuit in a way that it's stable, such that the cells on one end and the other are getting different information, and then when you alter that, it will keep. You can think of it as a flip-flop. The easiest way to imagine it is a flip-flop, because it has two stable states, and the other stable state is such that it will propagate to the other end such that both ends are head-like. So I'll show you how that works.
Okay, so can I quickly follow up on that? So you mentioned at the beginning the idea of these bizarre genomes that these animals have, right? And you said despite those genomes you can make a wild-type animal. My question is, are the incredible regenerative capacities and sort of new plans that these animals can store not so much happening despite a bizarre genome, but because of the bizarre genome? Is there a way to go about testing that? To what extent does genomic instability in somatic mutation propagation actually contribute to the regenerative capacity?
Yeah, that's a great question. I show that problem precisely because I think it's a fundamental, interesting problem that nobody has an answer to. No one knows. I mean, we're making models now of basically interacting cells in the same body that have different genomic information. It's a wide open field. It's not discussed at all, never mind solved. So.
I really love the idea of the self-propagating organogenesis and the beautiful, elegant studies with the planaria. One thing, though, I wonder about, though, is when you jump to, say, mammalian systems or other systems, why you focus specifically on the ion channels per se, because you could imagine the same self-propagating system occurring with epigenetic changes, with transcription factors, with other sorts of events which are self-propagating in the same way and perhaps fit into the mold, but not ion channels. And in fact, most of the literature on at least human morphogenesis would point towards some of those other things. And just to challenge you a bit, I guess is if the electrical signaling is so important in a, say, mammalian system, one might see this in chronic electrical stimulation, which is going on all the time with pacemakers. So there actually are kind of lots of interventions that are already being used, and one could predict that they would cause long-term changes. And at least to my knowledge, it's maybe it hasn't been tested, but it's unclear if that occurs.
Sure. Okay, two great questions there. The one question has to do with, I'll take the second one first, about what is the evidence for the importance of this in humans, and what about the electrical stimulation. So the kinds of electrical stimulation that occur in the most common medical use, so let's say electrical stimulation for muscles and wound healing and things like this, are generally not the kind of stimulation that will interact with this system. Rapid spiking, whether it be through a magnetic system or through controls of a peripheral nervous system, that kind of rapid spiking is generally not anything that's going to directly affect the system. There is, however, a whole body of data on a different type of exposure that human beings have, which supports these kinds of ideas, and that is the teratogenesis by channel modifying drugs. So a lot of the drugs that actually modify these kinds of events are in fact teratogens and are pretty dangerous, both in let's say rat or mouse models and humans. So you would not expect to see this with a kind of stimulation, magnetic field exposure. None of those things really alter long-term steady-state electrical properties of cells, but you do get this from certain kinds of drugs that interfere with this system. The other interesting question is, why focus on bioelectricity as opposed to chemical gradients? So I certainly don't mean to say that chemical signaling systems are not important. Absolutely, they're important, and all of these things that I talked about are no doubt the result of cooperation between electrical, chemical, and physical forces, pressures, strains, tensions, and so on. All of these things are important. The reason we focus on bioelectricity is because electricity is remarkably well suited for things like memory and decision making, and the reason is more so than diffusion. I mean, I've seen people create specific circuits out of diffusing molecules, and it usually ends up a very fancy paper in Nature and so on, precisely because it's extremely difficult to do this. Electricity is, I think, evolution discovered very early on that for the same reason that we make computers and so on electrically as opposed to through chemical diffusion, is that it's much easier to process information that way. And specifically, what I mean is a voltage-gated channel or a voltage-gated gap junction is a voltage-gated current conductor. This is in effect a transistor. It means that using circuits made of those things, you can easily build up to almost any kind of computational process that you want. And could somebody make models of all this stuff that purely were with diffusing morphogens? Possibly. I'm not going to say it's impossible, but I have to see it first to judge that. Whereas I think this is a really a tractable control system. I think a lot of the decision making, if you think about artificial neural networks, right, they really show you how memory and decision making can be implemented by electrical networks, and then of course they work together with all the chemical gradients.
Last question.
In the situation where you took the planaria and changed the head shape or structure, does that propagate That is either by cutting or from the progeny itself?
Right, yeah, that one does not propagate. So what happens, so unlike the two-head state, which is permanent, that one does not propagate. And what happens with that is they regenerate the head appropriate to some other species. They sit there for about 30 days, and at the end of 30 days, they suddenly, something suddenly kicks in where they decide, wait a minute, this isn't right, and they will remodel and go back to their original state. So that effect does not propagate.
Do you have any sense of why that memory comes back or why you can't cause it to persist? Have you guys sorted that out yet?
Yeah, we don't. Our best models are for the head-tail decision, not for the head shape. We don't have a great model yet for the head shape thing, but we do know one thing that's a little bit similar to that, which may give a clue. If you take there was a classical data that people would take a tail blastema from a salamander, and they would want to know whether that thing becomes a tail because it's locally determined or not. And they would attach it to the flank of that salamander. And the data were that in the short term, that thing makes a tail. And so you would say, okay, it's locally determined. But then over the coming months, it would eventually remodel to the more correct pattern. So it seems like, so maybe, and this is pure conjecture, maybe what we have here is a two-phase process where there's almost a short-term and a long-term memory that are distinct. Right? But we really don't understand that mechanistically at all, and so after we get through really analyzing the one-head, two-head thing, we're gonna turn our attention to that.
Okay. Thank you very much.